So our speaker is originally a Californian, one of your natives. This is Dr. Lora Richards. So let me give you a little bit of information about her. She was an undergrad at UC Davies, where she studied ecology and evolution. She got her BS there. And she had a minor in plant biology. Then she did her doctorate at University of Utah in Salt Lake, and there she studied tropical plant-insect interactions, and she got to, so I want you to listen to her career trajectory, because she's had a lot of fun. So she did tropical plant-insect interactions in Panama. Then she did her first postdoctoral fellowship. She had an international NSF postdoctoral award, and she spent 2 years in Australia, studying, I'm not a biologist, latitudinal gradients, which is something to do with looking at forests at different latitudes.
Yeah. Tropics, tropic, subtropic, temperate.
Yeah. But the good part about that is that she spent 2 years in Australia, and she had 2 cars there, one in Queensland and one in Tasmania. So you can imagine that was pretty fun. And then she came back to the States, and she did a second postdoctoral fellowship at the University of Nevada in Reno. And that's when she started becoming a chemist, because notice before that she hadn't been a chemist yet. And at her second postdoc at the University of Nevada, Reno, she studied chemical ecology. And for the last 2 years, she's been a tenure track professor there at the University of Nevada, Reno. Lora.
Yay. All right, well, I'm excited to be here. I got to visit my new colleague, Carolyn. So I'm coming from the University of Nevada, Reno, and I'm going to tell you guys a whole bunch of studies that I've been working on. And if you have any questions at any time, you could totally interrupt me, and we'll get started. So I'm looking at diverse phytochemical profiles. So like I said, I'm a biologist by training. I have ecology, evolution, plant biology, and then I started getting into chemistry and was super fascinated. I'll admit, as an undergrad, I did not like chemistry at all. So there's hope for some of you out there. You could still like it. All right, so getting started. Let's see. This was working earlier. Not working now at all. What happened? All right, this is a picture from my PhD work. This is a picture of a tropical forest, and I want you guys to look at this for a minute and just think about all of the plant species that you might see in this picture. Okay? You start thinking about all the different species you might see, then you can start to think about all of those bugs that might eat that plant. Okay? And knowing that those things are eating that plant, now think about all of the predators that are eating the bugs that are eating the plant, right? So if we start thinking about these taxonomic scales of this just revealed all my secrets. Embarrassing. Okay. We can start building these food webs, right? And we see all of these plants and everything, all these bugs are eating these plants and they're being attacked. And what I like to think about now, when I was doing my PhD, I was just thinking about all these interactions. But now, what I think about is I think about tiny little chemical profiles on all of these plants. Because what we know is that chemistry is mediating lots of these interactions. And this is particularly important because we're in the middle of the Anthropocene, which is like the 6th mass extinction experienced in time, right? So it's the 6th in time, but probably when it's all said and done, probably the largest as far as loss of taxa on the planet. And what we know is that what's mediating these interactions is the chemistry. So here is a picture of, these are plants here. These are the connections between the herbivores that are eating all of these different plants. And these are actually parasitoids that are attacking all of these herbivores, right? And so we could think about how the chemistry is mediating these interactions that are mediating these trophic networks. And when we think about something as simple as a plant, a caterpillar, and a parasitoid, we are encompassing a large proportion of all the species in the forest. So this is where I was fascinated. And when we think about chemistry, there's not just one compound or 2 compounds that are important, but plants produce this really suite of chemistries, right? These really diverse phytochemical profiles. And there's lots of different ways that we can get there. So I work on this species called Piper, or this genus, Piper, which I'll tell you a little bit more about. But it can create really, really diverse phytochemical profiles using different classes of compounds. So it takes lots of steps, or little steps, down multiple biosynthetic pathways to produce really, really different chemistries. That means that not all sister species are going to have similar chemistries. Or you could be something like alfalfa, which produces these diverse phytochemical profiles within a single compound class. So it has late functionalization within the biosynthetic pathway, and it kind of starts with the core structure and just manipulates it. And you can have all of these different compounds. So for example, getting 30 saponin glycosides, so one class of compounds. Both of them are super diverse. And what you'll notice is that we have not only can we get, oh, here we go. Here's a bunch of complexity when we think about chemistry. So I know that you guys have mixed majors, so some of you might see this and not be so excited like some biologists, but I am excited about it. And when we think about all of these structures, there are different levels of complexity we can think about. So we can think about compositional complexity. So how many compounds does a plant make, right? Well, how does it manipulate those relative ratios? Or it can have some structural complexity. So when we think about this, the alfalfa and those really complex saponin glycosides, what's that structure that's going to contribute to that complexity that we can pick up on? So this complexity is really important, and these mixtures are really important. And one of the reasons they're important is for synergistic effects. And so synergistic effects is where the effect of any single compound is greater than you would predict additively, okay? And so here's an example that you guys might be familiar with. So if you guys all wanted to go have some caffeine, for example, like I had several cups of coffee today, you have a lot of different options, right? And the caffeine that you're consuming in any of these beverages is going to be exactly the same, okay? It doesn't matter whether it was synthesized, whether it's from a natural product, or where it came from. It's caffeine is caffeine. But what's different is what's in the mixture of all these drinks. So if you were going to go get some mate, you'd get some other xanthines, similar in structure to that caffeine. And xanthines are known to stimulate the heart muscles, relax the smooth muscles, and you would have a very different experience than if you were to have coffee. Coffee has these monoamine oxidase inhibitors. Has anybody heard of an MAOI? Is there any pre-meds out there that might think about what an MAOI is or anything like that? Okay. Oh, you do?
It's a category of antidepressants?
Yes, antidepressants, right? That's why I'm so happy. I've had lots of coffee today, and I've had lots of MAOIs, which is one of the first antidepressants. Good job. And then if you were to go and get something like an energy drink or a soda, I mean, you're totally gambling with what that effect's going to be, right? Because there's a huge complex mixture in there that has been untested, right? So while we might experience these differences, so do caterpillars. So caterpillars can feed on different plants, and depending on that mixture, it'll have a different effect. So here's an example of a story I'd like to tell you. So here is Piper sinoclatum. This is a nice tropical. This one is actually found in Panama. And this is a really cool plant because it produces 3 of these amides. You see these here? And even if you're not a chemist, you can look at those and say they pretty much look the same, right? They look very similar in structure. And this plant has herbivores that attack it. And what's also neat is it has these biotic defenses. So it houses ants within the petioles and produces fruiting bodies for these ants. And when the ants are there, it produces these at kind of lower levels. So you might ask, why produce 3 of these compounds, 3 of the different compounds? Why don't you just produce one that's super toxic and does the job, right? Well, what happens is when the ants are not there, let's see if my animation works here. Let's see, might be a little bit out of order. Okay, well, when we look at these 3 different compounds, and here we did a bioassay with this Spodoptera. This is like the lab rat of insects. And we could feed it this casserole, so we put it on an artificial diet, and we could dose those diets with these different amides, okay? And here, red is the control, so this is survival. This is a percent survival over time. You guys can see that. And these are the 3 different amides, and red is control, and these are all at different varying concentrations. So we're trying to get some dose dependency here. We found that any single compound, they seem to do okay, but as soon as you put all 3 of them together, they all kick the bucket. So it's the combination of them, and it's not even just a concentration effect. So if you look at the concentration here, this is 0.1% of dry weight. This is 0.001% dry weight. So that means that even at the very, very smallest concentration, the combination of these 3 are much more toxic than any single compound, right? So when we're thinking about complex mixtures, the combination of those are really important. It's not just any one compound. So now when this plant doesn't have any ants on it, it actually increases the production of these 3 compounds. And we know that those are toxic for this generalist, but this generalist never eat a piper in its life. So maybe it doesn't really understand how to detoxify this chemistry. So what we did is we looked at we collected larvae out in the field, and we put them in a random assignment of treatments. And we either dosed the plants with a control, or we increased the production of any single amide. What we found is when we had all 3, the mixture of all 3, the parasitoid success was much higher. So more parasitoids, it made it more susceptible to natural enemies when it's feeding on this complex mixture. All right, oops. So when we think about plant defense, there's lots of different hypotheses out there on why plants produce these types of compounds, how they might allocate energy to these types of compounds. And we're not going to go through them, but most of the times when people are trying to answer these questions, they focus on their favorite compounds. So here's a meta-analysis looking at the different classes of compounds addressing herbivore performance. And so one of the faults that we've had in the past trying to address all these questions is we might look at these plants and say, oh, there's more of this than anything else. This must be the most important compound. So it's kind of a anthropogenic view of what we think is important, and we test what that's going to be. But now we have these different technologies where we can look at the whole complex mixtures. So we can actually take spectroscopic data, we can use chromatography or NMR, which I'll talk about a little bit, and use these new technologies or these new instrumentation, and the data acquisition is much more available to us. So now we don't have to make assumptions on what's important. We can let the data tell us. And so this is what I'm going to talk a little bit more about. So I have collaborators here in Boise, but I also have a huge team of collaborators that we have at the University of Nevada, Reno. And so to start tackling big questions, it's really important to cross-communicate between chemists, ecologists, people that know how to do population genomics, taxonomists, and biochemists. So I'm just having a shout out to my peeps here. And so I'm going to kind of talk about 3 different parts today. I'm going to go through community patterns, look at interspecific variation, and these are all going to be sort of buggy and tropical. And then I'm going to switch gears and do talk about some rats, some wood rats. So first off, I wanted to look at how does the role of phytochemical diversity play in herbivore diversity, herbivore diet breadth, multi-trophic interactions, and can we map these patterns on a phylogeny? Okay, seems kind of simple. So I really want to link, how do I take data like this and tie it to data like this? These are, for example, herbivores that feed on a single plant in tropical systems. So one of my favorite hypotheses is this hypothesis called the optimal defense hypothesis. So this is the hypothesis is that plants will allocate their resources and optimize that cost to defend against everything that's attacking them, right? And so plants are not just attacked by that one little caterpillar I happen to collect, right? They're attacked by pathogens. They may be attacked by mammalian herbivores, different insect herbivores. And so any given plant has to sort of defend against all these attackers, right? So the hypothesis would be that plants that had higher phytochemical diversity or chemical diversity would have greater biological interactions. And the reason I hypothesize this is because they're really trying to optimize that mixture against all those attackers, right? So to test this, I create a metamodel. This was a model that I would use to put into a structural equation model to get causal effects between all of these data. So here in my model, I thought phytochemical diversity would be an indicator of herbivore diversity, right? You're producing these big diverse phytochemical profiles just so that way you can defend them and not lose any leaf area, so affect herbivory. And also phototoxicity is one mechanism by which that can happen, and I just happened to find a paper that worked out well for that. So this is working on this genus Piper. So Piper is really cool. This is where your black pepper comes from. Okay? It is super chemically diverse. It makes all of these different compounds, and it's really not well defined. Because it's a tropical system, it's pan-tropical, it's old world, new world tropics, and there's it says over 1,500, there's about 2,000 species of this. So they're really interesting to study chemically. And there are these species swarms. So if you go into a tropical forest, there's a couple of genera that, when presented, they presented multiple species that fill the same niche, which is kind of interesting that they all are these understory shrubs that coexist right next to each other. And normally, you wouldn't want to exist next to congeners because you may share enemies, right? And we don't want to share enemies because that would be bad for us. So this is really interesting to see how the chemistry maintains these different levels of diversity within these systems. So we have these long-term ecological data. So working with the Earthwatch Institute, we have these long-term data sets where we go out in the field, we collect caterpillars. These are through the use of citizen scientists, so these are really fun trips. We collect caterpillars off the plants and we rear them out, and they either get this is a tachinid fly, so tachinid flies lay their eggs on the outside of that caterpillar. The egg hatches, and the maggot burrows into the body, and then when it's ready to emerges, kills the caterpillar, pupates into a fly. So nice parasitoid system. Or you can get other parasitoids like this wasp here, which will oviposit its egg directly into the caterpillar. So it's kind of cool to collect a caterpillar and not get a moth or a butterfly at the end. You get some crazy parasitoid. So we have all of these interactions so we could figure out which herbivores are eating which plants. And this is really interesting diversity data because most diversity studies just set out a light and just see how many insects come to that light. They don't really have the ecological context for it. So I wanted to capture the chemistry. So there's a couple different techniques that you can use to capture chemical data. And so here we chose, for this project, we're looking just at NMR. So looking at this really broad spectrum analysis, okay? And NMR, who knows what NMR is in here? Okay, there's some people that do, right, great. So NMR is telling you, it's like a big magnet, it's like a big MRI that you put your compounds in, and it's telling you that micromagnetic environment around each proton. This is proton NMR, and so where it falls out along the spectra is telling you sort of what that neighborhood is that a proton is in, and it tells you who it's interacting with through peak shape and the number of protons experiencing that same micromagnetic field. And it's really nice from the standpoint when we think about structure and compositional complexity, it can incorporate both. So, for example, a single compound, like this one right here, would present itself in multiple places across this spectrum. And what's interesting is that there are certain patterns on what kind of functional groups you would find at different areas. And so I was kind of stuck with this position where I'm trying to look at phytochemical diversity within a species that has no shared compounds across all these species. So I used structural diversity and compositional diversity through NMR by calculating diversity measures just from the spectra, just like you would for species diversity. So I can look at the peaks and look at the area under the peaks and just get an idea of how diverse these two spectra are. and use that as an indicator of diversity. And so looking at phototoxicity, I found this old paper which happened to have crude extracts of the exact same plants that I was studying, and they put them in a bioassay of 3 different bacteria, and they grew them in light and without light to see what the effects of inhibition were. So I can get a measure of phototoxicity. And something that is important is that if I'm focusing for the phytochemical diversity component, I'm focusing on the downfield region, which tends to have these, it's less crowded and has these really key functional groups. And you'll find things in that downfield region, such as these, this pattern of double bond, single bond, double bond, single bond, and you usually need that to be photoactive. So we found that phytochemical diversity was actually a really strong predictor of herbivore diversity. That means that those species that had high phytochemical diversity in their NMR profiles had more herbivore species, the herbivore community that was attacking it was more diverse. We also found that those species that had higher phytochemical diversity had lower herbivory, which is what you'd expect, right? If you're making all these diverse chemicals, you want to reduce herbivory loss. And we found that it was also a predictor of phototoxicity. Again, we're focusing on that downfield region of the NMR where you would find conjugation that would be photoactive. So that was nice. And that also was those extracts that were really phototoxic had were negatively related to that herbivore diversity. So this is a way, these are structural equation models that we can then look at the partial correlations. This is what the real data look like. So you could see the patterns. And so these are partial correlations, so they control for variance of other variables within the model to get more of a causal relationship between these variables. All right, so we found that phytochemical diversity did support or was a good predictor of herbivore diversity in the herbivore community. So next, I wanted to look at, herbivore diet breadth. So herbivores can either be really, really specialized, where they feed like only on a single species within a single species, or they could be something that's really, really broad, like this guy over here that can feed on 8 different pipers and 40 other plants, right? Super mobile, eats everything, doesn't spend its entire life cycle on a single plant. So we have this nice gradation of generalists versus specialists. And so when we think about toxicity in plants, we can imagine that if you're really, really toxic, your herbivore that can handle that must be really, really well adapted to that, right? So we might make some predictions that plants that have higher phytochemical diversity, which might be more toxic, for example, would have, the herbivores would have a narrower diet breadth. So they would be very specialized onto these really specific profiles. Or you would have to be somebody crazy, like this guy down here, and just feed a little bit on everybody, all right? And so taking the same data, we looked at the different levels of diet breadth, because we had this big long-term database. We can see of those caterpillars, what else did they eat, and we could get the diet breadth of each individual caterpillar found on each species. And we found that phytochemical diversity, that those plants that had high phytochemical diversity, had those herbivores that the generalists that fed them had on really, really broad diet breadths. So they could eat lots of different things, and that contributed positively to that herbivore diversity we measured. Alternatively, if we had phytochemical diversity had very narrow diet breadths. So those specialists that fed on them were single species specialists that could feed on that one species. And that also contributed to the herbivore diversity. This looks negative to you, but when you connect phytochemical diversity to herbivore diversity, that negative times a negative equals a positive. Great. So this is sort of a picture of what it would look like. So we would have changes in phytochemical diversity, and you can imagine that herbivore diet breadth would go from something that might be a little bit medium of the road to this really, really broad distribution of specialists versus super generalists. All right. So found support for that hypothesis that we were thinking about. But one of the mechanisms by which chemistry can interact with herbivores, like I showed with the amide and those synergistic effects, is through the immune response. So we wanted to look at, does phytochemical diversity then affect that parasitism rate at the other end? So this is working with my collaborator who does eco-immunology. And we have an assay by which we can measure phenyloxidase. And so this is a cascading effect within the caterpillar that is an indicator of potential immune response. So you can challenge the caterpillar, or you can also measure that immune response of the phenyloxidase as a precursor to its potential ability to defend itself against a parasitoid. So we wanted to look at what the bottom-up effects of phytochemical diversity on parasitism through this immune response. So we found that in phytochemical diversity, actually, remember, we saw this before, that plants that were super phytochemically diverse had specialists on it. And when you think of a parasitism and parasitoids that are living inside of a caterpillar's body have to be pretty specialized, because you have to figure out how to deal with their immune systems, their organs, everything in there. So they tend to be, so those that had very, very narrow diet breaths with higher phytochemical diversity, with narrow diet breaths, also had higher parasitism rates. So those specialists had higher parasitism rates, and it actually affected immune response. So the phytochemical diversity had a negative effect on their immune response. So if they're feeding on a plant that was really diverse, they had suppressed immunity, which then also, when their immunity was low, their parasitism was high. So it's kind of cool. We were able to link not only chemistry to herbivore communities, to the herbivore diet breath, but also to these multitrophic interactions of how it affects parasitism. All right, so we found these multitrophic effects. So this idea that plants produce these toxic compounds, you might expect if you have this like arms race of being able to get something toxic and then an herbivore adapting to it and then getting more toxic, there's this hypothesis that you would get this defensive escalation over time. And here's looking with a classic, this is where you would expect that more derived plants would be more defended or more toxic than ancestral plants, okay? And we've seen this in Bursar. This is a shrub found in Mexico. Over time, this is millions of years, they have an accumulation of species, and they also get an accumulation of compounds over time. And this is really interesting because these are all terpenes, which have a shared biosynthetic pathway, okay? But we didn't find that in our plant system, because when we're looking at phytochemical just diversity itself and the measure that we're using, and these plants produce everything under the sun, we didn't find any of these patterns, but we wouldn't predict it within this system. Maybe we could have predicted it in a different system that uses all of a single class of compounds, perhaps. So we did not find any support of any phylogenetic patterns here. So I've been talking about chemical diversity on a single scale. This is looking at it within a single sort of group of, just as a species measure or a single spectrum measure, oops. But there's another level of diversity that incorporates sort of change or turnover or chemotype. So how plants are different within a species across the forest. And this has big implications on diversity as well. So for this, we looked at, we're going to look interspecifically. So a single species of piper, do we see similar patterns? And we're going to look at how the abiotic and biotic consequences of phytochemical diversity and herbivory across this landscape. So this is Piper kellii. This is actually, we've now moved to Ecuador. This is in the Andes, nice cold Andes, and they have these piper. This is the piper right here. You can see all of these little windows shadowing here. You guys see all that herbivory right there? This is all from these Eoas. So piper is really interesting because the whole genus Piper has a whole geometra genus that has radiated on it. So the whole genus only eats piper. And this species here is called Piper kellii. We call it Piper pink belly. It has this pink coloration underneath the leaves. And there's like 10 species of this Eowys that feeds on this single plant. 10 species of a caterpillar that feeds on one plant and not found anywhere else. So we've done the chemistry on it. We know it actually produces these 3 major secondary metabolites. And what you'll notice is that you see this pattern again, single bond, double bond, single bond, double bond. So that should give you an indicator that there might be some photoactivity here. We also have done the, so this is one caterpillar. We did genotype by sequencing. So we looked at the genetic structure of this caterpillar across the landscape. And what was interesting, so here, this is a PCA analysis. And so what this is telling you, each one of these triangles is a caterpillar. And how close they are is how closely they're related genetically. They're genetically closely related. And what we found is that when you survey up a mountain, that all of the ones up the mountain were different up there. And when you overlay the chemistry of those plants that those caterpillars were found on, the chemistry is really different. So it had more of this prenolated benzoic acid up at the top of the mountain. So it's really cool. So we had a grad student. Oh, wait. The other cool thing is that when we think about up a mountain, we can think there might be changes in light. So you get different exposure of UV and when you're at the top of the mountain versus the bottom of the mountain. So working with a chemist, we have a chemistry collaborator and a photochemist. We could take the extract of these plants and we shine a light on it. Remember how it's called pink belly? It's kind of pinkish. When you actually radiate light on it, you get this sort of long-term color change within that chemistries. So we know, so we have some ideas that these might actually be photoactive compounds. We also took the compounds and dosed these artificial diets and fed them to this Spodoptera, this lab rat of insects. And we found that here's control, and here's, we put some of the plant material in there, and under different levels of UV, with and without UV, you can see that the mortality, they all died when you added UV light compared to control. So we know there might be some phototoxicity within these plants in the system. So working with our grad student, Andrea Glassmeyer, we went and she set up these paired experiments where she threw up a rope and Piper can grow by cuttings. So you can get like all identical plants. So we were able to get all of these cuttings, and she had some at high levels and some at low levels. And so you can imagine higher in the canopy, you can also get more light up there. And she went all the way up the mountain and did a bunch of these, set a bunch of these up, and then went back, harvested them, looked at the herbivory to see what happened. And so one thing, so she did NMR and she did LCMS on this. Okay? So with the NMR, she found that light had a big effect, or the canopy height had a big effect on phytochemical diversity. So just the exact same plant, higher in the canopy and lower in the canopy, had totally different chemistries. Okay? So phytochemical diversity was different. And she couldn't get the light measurement way up there in the canopy. So, but she did take hemispherical photos of the lower one, so she was able to get some measure of light. She found that, that when she had, if you look at the amount of light, that as the light increased, the phytochemical diversity decreased. And this was kind of interesting. And so what we did is we then looked at the mass spec data. So this is just NMR, so we're looking at protons across the spectra, and now we're looking at mass spec, and we could do a factor analysis to try to figure out of all these peaks of the mass spec, what's the difference between all of these plants. And she found that it loaded between, there's this dichotomy between producing this piplartin, which was kind of cool. This, you recognize this shape? You recognize this? This is like the amides that we saw, those 3 amides on the other plant, compared to these chromine pathways. So those are those chromines, remember, that have all of those potential conjugations that are making it photoactive. So we could then put this in another path model and see that we can get canopy height is contributing to this chemotype. So whether they're producing more of these chromines, those photoactive compounds, or these amides, depending on the light they're exposed to, that's having an effect on that NMR reading that we're picking up. And that has a really big negative effect on herbivory. Both of them, this had a negative effect on herbivory. The chemotype itself had a positive effect on herbivory, but also elevation had a big effect on herbivory. So we're trying to now get together and pull together all of these different abiotic and biotic factors to really figure out what's going on with this cool plant across this landscape. But what we're continuing now, so this paper was published this year, in the beginning of this year. But what we now have is we have all of these plants that have all these different exposures to light across an elevational gradient. We didn't find the predicted difference in chemistry across elevation. There's actually no effect. It was mainly this high and low canopy. But because we have all of this nice variation, we're now leveraging that to create these biosynthetic models to get some ideas of how we can make predictions of these intermediates and some of the biosynthetic pathways that we might predict from these plants. So we've now got vats and vats, all of the little peaks of the mass spec. Our chemists have isolated and working on structure determination. So it's kind of a really, really cool way to look at chemistry. All right, we're now moving into vertebrates. So I've been talking about this tropical systems, super diverse, lots of species, but now what does plant chemistry and the maintenance of species have to do when you move to an environment, like this? Okay, this is down in Southern California. This is the desert here, and this is an area where it's very flat and dry here, and you have this hill, and you see how there's more green on this hill? It's a little bit more mesic, has a different vegetation type. And there's these two wood rats that live there. Aren't they cute? Wood rats are cuter than other rats because their tails are furry, and I think that's a fact. And if you look at them up close, they're just adorable. And they feed on two different plants. So what happens is when these two wood rats get together, they can mate, they can make babies. The babies do okay until they get off of mother's milk. When they start getting on the adult diet, they don't survive, which is kind of crazy. And so what we thought was, well, maybe it's because there's these two different food types that these wood rats are feeding on. So we initially collected a bunch of poop, and we took the poop, we sent it off to do the DNA to see what plants are in the poop. And we do. We see that they eat totally different things depending on which environment they're in. And so maybe when they're hybridizing and they have their offspring, mother's milk is okay, but when they start to feed on that plant-based diet, unless they have the proper machinery to detoxify these compounds, then maybe that's what's affecting their survival. So, two different plants. I'm going to tell you about the chemistry of these two different plants. So this guy feeding on prunus, right? These are known to have cyanogenic glycosides. Okay, cyanogenic glycosides are pretty toxic. Has anybody had cassava? Nobody's had cassava. Okay, cassava is a dominant food in places like Brazil, and it has high levels of these cyanogenic glycosides. Cyanogenic glycosides by themselves isn't super toxic, but what happens is as soon as you cleave off some of these sugars and you get this hydrogen cyanide, super toxic, okay? 0.5 to 3.5 milligrams of hydrogen cyanide per kilo body weight can kill you. People who eat cassava have to boil it for a very, very long time. You can't just go to the grocery store and eat it like a potato, okay? So super toxic compounds. One wood rat is eating these super toxic compounds. The other one is eating this rhamnus that is known to have anthraquinones. And anthraquinones are known to us as laxatives, okay? So one of the dominant compounds is this with it is this anthraquinone. See, this has a sugar moiety on there. So when this emodin comes in, gets into your intestines, right? Gets absorbed, blocks sodium potassium pumps, alters water retention, increases peristalsis, right? So it might be nice if you have a short blockage. Maybe you might want to hit one. But you don't really want to be putting a lot of this in your system because it really, really does mess with your intestines. So both of these organisms are eating these two super toxic compounds that have different modes of action that might be affecting the way that their offspring are surviving. So we went out and we wanted to see, is this really the full view of their diet? So these wood rats are super convenient because they have a latrine. So they do their business in one spot all the time. So we had Danny went out, our grad student went out and he cleaned their latrine and he waited 3 days to get some fresh sample and collected it and brought it back. So we have all these fecal pellets. Then we could do the chemical profiles on those fecal pellets. We can look at the microbiome and the plant diets and look at the tRNL to see what plants are they eating, what's the microbes that are coming out, and what does the chemistry look like. So we did this for multiple times between the two habitats, multiple times a year. So this is what it looks like. So these are round, they look like pellets, and they represent pellets. So these are each represent the fecal pellet. This is LCMS data. So in LCMS data, you have all these little peaks that represent compounds. And so the way this network works is that each of these pellets that are connected is because they're chemically similar, that those chemistry out of each of these poops are more similar to than if they're not connected. And what you find here is we have it mapped out where these are the hills, the flats and versus the hills, right? The two species that we see. And you can see this weird glob where they're both together, right? So if we then want to map now, look at the seasonality on top of it, I can color code it for when Danny went out and collected those poops. And so this is kind of interesting. So you can see that they have very, very specific diets. So what that means is that the chemistry of their fecal pellets coming out are unique most of the time of the year. But in May, they actually converge onto, and their poop chemistry comes out very similar. And what we found is that they actually are feeding on, once they're all feeding on this forb that grows all over the up and down the hills and the flats that seems to be super nutritious during the spring, right? Which could be really important if you think about breeding seasons and all of these sorts of things. But there's something here that should probably stand out to some of you guys, if you were paying attention, would be this Magenta Doll right here, right? Like what the heck's going on in December? So in December, we were only able to visit one of the sites. Danny wasn't able to get, you'll see that December is up here. So he only was able to get it in the flats location. He was able to go, it was sort of opportunistic sampling. But what's cool is that wood rats are pack rats. And so while this may not be like statistically viable, I think it's really cool because this means that they were probably storing some of that plant material and they started feeding on it in December. And that's why their poops look like May pellets, because they're probably eating similar plants that they're eating in May that they had stored in their middens. So I think it's really interesting that we can take chemistry, just take some poop samples and look at the chemistry and look at the plants in there and see what's going on. So if we also then take these poop, I guess I have that there. Okay. We can also look at these clusters and try to map them on and look at which microbes and which plants are associating with clusters of different compounds in the chemistry. So here is another network. Now instead of each thing being a single fecal pellet, it's a single mass within the chemistry, the chromatograms. And you can see that they cluster, and now we can get associations with particular parts of the fecal chemistry that are associated with particular plants. So here's Eriogonum, or here's that prunus we were talking about, or the, where's the rhamnus? I guess I didn't have it up there right now. and particular bacteria that are associated within the microbiome that might be helping them to detoxify some of these compounds. And so, with all of that, maybe when you go out in the forest or your local environment and you see all of these plants, you'll think about how complex all of these interactions are and how chemistry is at the root of it all. And that's it. So I'll take any questions. Yeah.
So I know you said that you didn't really much care for chemistry in undergrad.
I did not.
And then you found this love for chemistry in graduate school.
Yeah.
How was this different than like your experience with undergrad, and how did that
Okay.
change?
Good. That's a great question. So I think that I went into chemistry thinking I was going to be bad at it. And so when you kind of go in and go like, I got to take chemistry. But I think what's different is that now I'm wanting to learn. I want to learn about it. And so I think just putting it into a context rather than just sitting like in my OChem class, just having to memorize synthesis steps for something that I couldn't. It just seemed like I was going through just making somebody else happy. And so once you figure out, put it in some perspective, something that is interesting, then it got super interesting, and now I just love it. So, yeah. But I'm not a chemist. I work with chemists, interpret chemists.
Do you have any suggestions for people who are going to graduate school, what they should be doing at this stage?
How many people want to go to graduate school? Wow, okay. Yeah, so have you guys been starting the process of, do you guys know how to start that process? Aside from just doing the GRE? Beyond the GRE, right? So what's important for graduate school, and it's different for different fields, is to figure out what the hell you want to do in graduate school, right? What kind of, not just like, oh my God, I want to go to graduate school in biology or ecology, but it's like, what kind of questions gets you excited? And then find those faculty that do the things you like to do and reach out to those faculty. So when you send in your application, if you haven't contacted anybody at that school, it's hard for them to know who you are or what you're interested in or how dedicated you are. So if you can reach out to them and say, I really love your work on this, I'm interested in pursuing stuff like this for my PhD, then you can start that line of communication, and then that way, when they see your application, they're like, oh yeah, I met this person's awesome, they seem really interested, and you flattered them because you told them you thought their work was good, right? Because they're actual humans, and they love to be flattered as well, and so it's really important to contact those people and make that connection. And it's also really important for you to make sure that you can get along with that person. Because when you meet, you want to have a meet, whether it's a Skype or whatever it's going to be, you want to have that meeting with that potential advisor to see if your personalities mesh. Because some of these people can be as awesome, their papers can be amazing, and then you get face to face with them, and you're like, I can't work with you for 6 years or 5 years or whatever it's going to be. So it's really, really important to make sure you have a good vibe with that person. And it sounds cheesy, but lots of people fail at their PhD because they don't have a good relationship with that advisor. And so having a really good relationship and knowing how to communicate and knowing what kind of mentorship you need is really, really important. So, and it's also important, this is what I find, nobody wants to say that they need to be micromanaged, okay? Because micromanaging seems to have this negative connotation. Like it may make you think like, well, I can't work independently if I say I like to be micromanaged. So you don't need to use the word micromanaging, but if you are somebody that has a hard time finding structure, because there's not a lot of structure when you first start your PhD. There's like, oh yeah, you're gonna take this 1 or 2 classes, go figure out, depending again on the lab you get in, figure out what you're gonna do for the next, whatever, 5 years of your life in researching. And so there's not a lot of structure there. So making sure you can communicate with that advisor, that they can give you the right feedback that can help you be successful, is important. So they run the spectrum. Some advisors are super hands-off, and some advisors are way more hands-on. And so trying to be able to know what you need and finding that advisor that can offer you what you need is really important. And it's not something that, like, if you're like, I would like a lot of structure in the beginning and somebody to help me with that structure in the beginning, I think that's a totally fine thing to say, but it's good to be honest with yourself and with that person, because I've seen lots of advisors that are super hands-off, like not talking to their students, for weeks on end, and those students have a hard time sometimes figuring out what the hell they're supposed to be doing. And so just making sure you have that nice open communication is really, really important for when you're choosing your advisors. So it's sort of a double interview. So if you're really good, they'll want you, so it's not a big deal. So it's not like they all have like millions of amazing students just knocking down their doors. They're all ready for, good students who are super motivated. So anybody have specific questions about the process? Yeah.
How do you find research groups, like in different universities?
Right. So I would search I would do more primary literature searching to find those people who are doing the stuff you like, and then figure out which institution they're at. So there's a couple of ways that people do it. Either you go to some place that seems convenient for you, and you go see who's at that institution and see if there's somebody you might want. But if you could go after, because you're doing research, like that's the meat of it. So you want to make sure you look at the research that you find interesting, and then find those people, see where they're at. And there'll be a long list. So check that senior author, because that's usually, or some of the more senior people in there. Sometimes the first author will be not the person who has the lab. But that would be a good way. And then, especially if you start doing literature search of the things that you're really interested in, you'll start seeing the same names popping up multiple times. And that's when you could start figuring out who to talk to. And then even if you talk to them and they're like, oh, I'm not taking anybody, they can often refer you to somebody else. So I would start with primary literature search. That's how I did it. That's how. Then when you do it that way, then when you're contacting that person, they know that you're interested in them, not because they're convenient or they're at this fancy school that sounds cool, but that because you're truly dedicated to that research question. And that's what really makes you more successful for grad school, is being passionate about the questions. So.
Question, Mika?
Well, I don't know, did you ever feel conflicted about, choosing to follow, a science path? Or have you always been, a super hardcore, science?
So when I was in high school, when I was in high school, I thought I wasn't good at science at all. And then I took physiology and I fell in love. And so then I thought, well, I'll be a doctor or I'll be a nurse. And then I took a zoology and botany classes, and I realized I just realized I love science at that point. So I was always into sort of ecology, biology. And so but I didn't think about grad school until I had a research experience. So I did a field course, and then I stayed on in the summer and did research. And that's when I really, really felt like this is what I'd love to do with my day, is answer cool questions. Because that's what I think is the best thing about research, is that you get to ask questions, and then you get to answer them, which is you have to be into it to want to do that, because it's not easy to answer your questions. So, yeah. But I have not. You have conflictions on staying in science, or?
Yeah, I just don't know what I want to do.
That's okay. And if you try different experiences, I think that's the key, is just try different things and then see what fits.